In lecture 5, we're going to discuss how neurotransition occurs within a neuron. Before we get there, though, we're going to start talking a little bit about kind of the history, a little bit of the history, about how we have come to realize that the nervous system uses a change in polarity or electricity kind of current when it functions or when it's communicating. So brief history again, just touch on. I'm sharing this with you not because I want you to memorize every person's name and what they did, but I want you to kind of see. We take for granted, I think, today about how much we know, and we were talking, just a few hundred years ago, we really didn't understand anything about the way the body worked, and so this, even though this might seem like, well, no duh, now it's pretty astonishing how things were figured out. So, anyways. Galvani, if you're in any science class like chemistry, you've heard his name before. But anyways, he is responsible for recognizing the role that electricity plays in muscle contraction. And he did this because he worked with I believe it was a frog, and he caused the muscle to twitch when he used an electric stimulation on the nerve. Therefore, he concluded that electricity was how the nervous system, the nerve, communicated with the muscle fiber. Then we continued to move on. You had Catton, who ended up actually measuring electrical currents in the brain. First he did it on animals, and he basically used a voltmeter to start looking at the changes of electricity through the skull. And then you just kind of have another series of scientists who continue to kind of build off of this idea. And then in 1874, we actually had the first, I believe it was an anatomy, so a physician who started to test these things in the human brain. And then you have people coming in who are like, well, it doesn't, if it's electricity, it needs to move at a certain speed. Well, obviously nerve conduction does not move at the rate at which electricity actually flows, which is why electricity or a lightning bolt kills us if we have too much stimulation. However, they did start to measure, and they found that nerve conduction flowed at about 30 to 40 meters per second, which is dramatically smaller than electricity, but still it has the properties of an electric wave that is being passed. So it's not like a bolt or a charge release like lightning or the electricity out of your AC current wall plug, but it is a wave that kind of travels down the axon, and that's what we're going to talk about. This wave, here's a little picture to kind of make you think about it, is basically what an action potential is. So we're going to look through that using a caricature. So you got the neuron sitting there, right? You've got your dendrites out here, which are receiving the information. This neuron's receiving that message. You should have an action potential. And we've got this neat little voltmeter who is hanging out here on the axon, and we're going to watch what happens as this wave of electricity, basically it's a wave of ions passing the cell membrane. When we see that, we'll see it pass down and what happens to that voltmeter. So the passage of those electrons along the axon ends up finally reaching the voltmeter, which goes crazy. It is there and it passes, and then it goes back to normal. It's basically what we see happening, and that's the voltmeters are able to pick these up. And one of the places where we really started to study this was actually with the giant squid, because their axons were much larger than ours or other mammals that you could look at. So we really use the squid as an example to really start to understand the properties. So this is something that goes across multiple types of species. This nerve conduction, it's not unique to mammals, but it is how the nervous system seems to be communicating. Well, one of the important pieces before the message can even be sent is that the membrane or the resting membrane potential needs to be maintained. So we're going to talk about the major factors that play a role in how this membrane potential is created. Here's a picture where we've got an example that shows you those ions. If you go back to that last review that we talked about, we discussed the role of positively charged ions, sodium and potassium. We also talked about the one negative. Do you guys remember what that is? I'll let you think for a moment. There was one negative or anion. Okay, somebody might have gotten it. It was chloride ions, yay. So there are cations and anions. Cations are the positives, that's the sodium and potassium, and then the anions are the negative, is the chloride. But we also have negatively charged proteins, which are really large, and they do not pass the cell membrane. So when we're talking about the flow of ions across. We don't talk about the proteins going negative, but they play a major role in that in general, the cell, the neuron, is a negatively charged entity because of all the negatively charged protons or protein molecules. All right, so what are some of those principles? We've got the ions that we talked about. One of the things that we need to understand is that the concentration gradient plays a major role. And this goes back to chemistry and biology. It doesn't really matter, biochem. But it's basically the philosophy that ions, anything, any solution that is concentrated likes to go from high concentration to low concentration. For example, this ink drop here, it's really close together. What it's going to do is it's going to continue to diffuse and separate, and they're going to bounce against each other until they reach equilibrium right here. So that's what a concentration gradient is. That's one of the principles we need to have, or needs to be present for neurotransmission. Then we have the voltage gradient, which sounds similar, hopefully, to concentration gradient, but it has to do with charge. So let's think about this picture with the water and putting in salt crystals. So as salt dissolves, or salt water starts to integrate itself with the pure water, what's going to happen is the ions are going to bounce against each other again until they can get to that point where they're at equilibrium and they're the same distance apart from each other. That is what solutions like. They like to reach an equilibrium where they are equidistant from all the other components. One of the third principles for neurotransmission is the selective permeable membrane. So you remember we talked about this last class, we were talking about the neuron. What is the selectively permeable membrane? It's the cell membrane. Good. What we're talking about is the fact that this cell, we're going to pretend that this is a cell membrane here, and it will not allow ions to pass across it. So what you see is that the ions over here get to equilibrium only on the left side, but do not actually get over here because they can't pass through. So do you remember what neurons have, or what cell membranes have in them to allow ions to pass through? Some of you might recall. Good job if you said channels, ion channels, or pumps, or those types of things, and those ion channels can be gated or they can be just open. Here's an example where you have channels that will allow the chloride ions through. So what happens is the chloride ions pass through, but the positive ones cannot. So you see that the chloride ions, following their concentration gradient, quickly equilibrate where you end up with 5 on each side, but you'll notice that there's more positives over here, right? Because the positives can't cross. So then there's this pull back from the voltage gradient, pulling those chloride ions back over to the other side. And what ends up happening is some do go back, and there's still a differential in the charge because obviously it's going to be more negative over here. So look, see all these positives line up over here, right? Our nervous system uses the same type of processes to facilitate neurotransmission. Here we're looking at the resting membrane potential, and we see that there are 4 particles that we're talking about. 2 of them are negatively charged and 2 of them are positively charged. What are things that you notice? One, you see that the negative anions, these A stands for amino acid or protein, so these are the proteins, and you can see that they're heavily in greater concentration inside the cell. These are really large. They're never going to cross. There are not many protein channels, or there aren't many channels to allow them to cross back and forth. So they're really a non-player when we're talking about neurotransmission, but you have to recognize that they are present, and they are what create the neuron to be in a negative state at rest. Our other 3 players include the chloride ions, and you can see that there are less chloride ions inside the cell than there are outside, therefore setting up a concentration gradient. And then here you've got your sodium and potassium. Now these 2 have the same charge, but what you'll notice is that their concentration, so voltage gradient may not necessarily play a role right now, but their concentration gradients do. Potassium, based on their concentration gradient, wants to be where? There's more inside, so it's higher concentration inside, so it wants to move outside the cell. And sodium is the exact opposite, where there is more outside the cell than there is inside. So it desires, or yearns, or whatever, if we can make it anthropomorphize it. But it wants to follow its concentration gradient to go from high to low, which would be outside to inside. So resting membrane is negative 70 millivolts. Some of the videos that you will see that I share give a different value for what resting membrane potential is, but for the sake of this book and for this class, if you are asked that question on a quiz or on an exam, it is negative 70 millivolts. How is this maintained? Well, first you have your selectively permeable membrane, which keeps the large negative charged proteins inside the cell, and it keeps sodium outside the cell. Then you have the concentration gradients that are created, as we showed you a couple slides ago, where you have more potassium inside than outside, and you have more sodium outside than inside. So those concentration gradients are really important for a part of how we create this resting membrane, so that when we get the signal to have an action potential, it is lickety-split because of these forces that are occurring. Then you have the voltage gradient. one of the things that we have at present in the membrane at rest is that there are some ungated potassium channels that are available to allow potassium only to move in and out, back and forth. So go back to thinking about that sodium and chloride, where the chloride ions, more of them crossed over because of the concentration gradient and came to equilibrium there. However, what happened was the voltage gradient was not equal. So some of the chloride ions got pulled back over to the left side. This is kind of what's happening with potassium. It will come in and out of the cell because it wants to go out because of the concentration gradient, but the cell is so negative that the positively charged potassium ions want to get in, and they will come in and out. Very cool little video that you should watch. It really talks you through all these pieces and helps you visualize it, as talking can only do so much. And then the last part of the resting membrane potential is the role of the sodium-potassium pump. It does exactly what it sounds like. It pumps sodium and potassium, and because it's a pump, it's doing it against the concentration gradient. It is what helps create the concentration gradient, and we'll talk about that.
At the resting potential in a neuron that is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus changes the permeability of the cell membrane to sodium and potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal called an action potential that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close depending on voltage changes across the membrane. When no nerve signals are being transmitted, these channels are closed. A stimulus causes voltage-gated sodium channels to open, and sodium ions rush into the cell. The cell becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The cell returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. In the wake of the action potential, potassium leaves the cell, restoring the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting potential distribution of sodium and potassium ions. Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
Right now. All right, so here are the key players in neurotransmission. When we're talking about the action potential occurring, you have some ungated potassium channels that are present. You have gated sodium channels. You also have gated potassium channels. And then you have this sodium-potassium pump. And what it does is it actually pumps 3 sodium. So this picture is reverse of what all the other ones have been. Intracellular is here. So it pumps 3 sodium out of the cell and brings 2 potassium in. So it's creating a net what? It's creating a net -1 for inside the cell. Again, what it's doing is helping support that -70 inside the cell. So here are some important concepts or key words that I need you to understand as we get moving along, and you're going to hear them over and over again. We have hyperpolarization, and that is where the resting membrane potential goes from -70 to even lower negative, -73, -90, whatever. It's becoming more negative. It's hyperpolarizing. It is becoming more negative. And this can be done by two ways. This can occur because potassium ions can efflux out of the cell, or chloride ions can come into the cell. Just so you know that this picture is off, this is actually happening out here on the dendrites. It's not happening at the axon. Just a little side note. Hyperpolarizing means more negative and less likely to have an action potential. Depolarizing is the exact opposite. This is where you go from negative 70 to something closer to 0, or becoming less negative. This is typically occurring, this happens when sodium starts to rush into the cell. This is considered becoming more positive or having that go message. Now when we're talking about the action, so those are the two messages. Remember we talked about how neurons can only send two messages, they can only receive two messages. That's hyperpolarization, the stop or inhibition, and then depolarization, which is the positive or go message. So what is an action potential? This nice little figure right here shows you exactly what it is. It is a change in polarity. It's a brief reversal of polarity. And you go from negative 70 at rest. If you make it to negative 50 and hit threshold, you're going to fly all the way up to positive 30, and then the cell is going to recalibrate itself. It will actually overcompensate and hyperpolarize, and then it will go back to its resting membrane at negative 70. This is something that occurs in approximately 1 millisecond. Once threshold is reached, it happens. There's no turning back. It's like a toilet being flushed, unless of course you block it. But once you flush a toilet, if it functions well, it will cause water to go down. And that is when you reach negative 50. Again, in this class, in this textbook, you're going to use negative 50 as threshold. Videos that I've shared and other sources that you look at may have a different value. But again, on a quiz or an exam, threshold is negative 50. Once negative 50 happens, then all the sodium channels open because that's what they're sensitive to, and it creates that high, quick rise up to positive 30, and then the potassium channels that are voltage sensitive or voltage gated will then open and allow potassium to go rushing out. So let's review this real quick. I think this is one of the most important pieces, and it's trying to pull everything together. So what we have here with the action potential is at a basic state at rest where nothing's happening within a neuron. It's sitting at negative 70. That's what this is right here. And when it's sitting there in that state, the voltage gated or voltage sensitive channels are closed. That's what rest is. The ion channels are closed. There's no ions passing back and forth. Then the cell receives some information, and what happens if we're trying to get to an action potential, we're saying we're getting that go message, we have sodium that comes in. Now this sodium can be, sometimes students really struggle with me a little bit about this, because they're like, well, where's the sodium coming from? Blah, blah, blah. And at this point, what I just need you to do is we'll circle back to that, because really this is a cyclical process, and we have to pick one place to start. So I've picked talking about how an action potential is reached and how it occurs, and an action potential occurs down the axon. So we're talking about what's happening in the cell body because of what's happening out in the dendrites, right? So your dendrites are receiving messages and sodium is being allowed into the neuron. What happens is, what happens is the sodium channels start to open, which allows sodium or positive charged ions to flow into the cell, into the neuron. If threshold, which is negative 50, if that's reached, then all your sodium channels in that area open, and you get this mad rush of sodium. Why do you get this insane rush of sodium? Because sodium, if you remember, go back to the concentration gradient, sodium is more concentrated outside the cell than inside the cell, so it wants to move into it, and it's negative 70 inside the cell. So what does that mean? It means that the positively charged sodium has both concentration and voltage pulling it into the cell. It is very quick. It moves very efficiently. Now we get up here at positive 30. This is the height of the action potential. It always reaches this. If you get to negative 50, you're going to always get a positive 30 response. What happens at this point? Well, at this point, you have at the height of the action potential, you have the sodium channels close, see here, and you have the potassium channels open. So there are potassium sensitive gated channels that will open when you reach the positives. It's roughly just over 0, but they'll open at that point. And what happens is now you have potassium doing its job. So let's talk about potassium. Where was potassium in the resting membrane? Where was the greatest concentration of potassium? It was inside the cell. So you've got tons of potassium inside the cell, and when the channels open, the concentration is going to push it out. What is the voltage now doing? So all that sodium rushes in. It's now positive 30 inside the cell, meaning that outside the cell, it's what? It's negative. So now again, both forces are pushing the ion to move in the same direction. How cool is that, right? So the system is designed perfectly. So if the potassium rushes out, it overcorrects. We get into this what's called a refractory period, which basically allows the action potential to go in one direction. This overcorrection here, this hyperpolarization, allows for that information to happen, or for that directionality for the action potential to happen. And then what ends up happening is the potassium channels will close, and then the sodium-potassium pump has to do its job. Now, why does it have to do its job? Why does it need to pump sodium and potassium across the cell membrane? Well, right now, after the action potential occurs, what is at a higher concentration inside the cell? Sodium is. And potassium is a higher concentration outside the cell, which is the exact opposite of resting membrane potential. We need to get back to resting membrane concentrations so that we can be ready for the next action potential. Now we're at the negative 70, so we're at the right polarity or the right voltage, but this is where the sodium-potassium pump starts taking 3 sodium, removing it outside the cell, and then bringing 2 potassium in, and that's going to recreate the concentration gradient in the way in which we need it for resting membrane. All right, that's the end of this section. I'm going to leave you thinking about that. I have 2 videos that are, one is designated towards just talking through the resting membrane potential, and another one is just talking about the action potential. I suggest you review both of those. They are videos of me drawing on a whiteboard, talking through each point, and they're really, really important. This is, when we talk about what material is really important, this is it, because these are facts that you need to have under your belt as we move forward, because we'll be talking about action potentials as the semester goes on.